Okay, welcome. We are going to be talking about chemicals and cancer here in Chapter 5. And I have this lecture divided up into 5 parts. So first I'll give just a brief intro history in terms of the discovery of the fact that chemicals can cause cancer. Then we'll talk about some of the common trademarks of chemical carcinogens. We'll look at, well, what are some of the common sources of exposure to chemical carcinogens? And then I will briefly, in the second half of this lecture, the second video, go through some of the 5 different key classes of chemical carcinogens. And then at the very end, we'll look a little bit at mechanistically how do chemicals cause cancer in a bit more detail. All right. So let's start by asking, what does it mean? We say, okay, this chemical is a carcinogen. This chemical causes cancer. That does not mean that exposure to that particular chemical is definitely going to give you cancer. It just means that exposure increases your risk, right? And that can vary pretty dramatically over a broad scale. So for example, you've got things like aflatoxin, where exposure to a very, very tiny dose will likely give you cancer, versus something like saccharin, artificial sweetener that I'll come back to, that we humans still consume today, right? It's approved for human consumption, although it is technically a carcinogen. It's just it would require a very high dose of saccharin in order to give you a significant cancer risk. To kind of tie this back in with some of our previous discussions of cigarette smoking, you're probably aware of people that have smoked for a very long time and have never gotten cancer versus somebody who, maybe smoked just a little bit or didn't smoke at all and still got lung cancer. And again, that really comes down to just increasing your risk of exposure. So the more you smoke, the carcinogens in that tobacco smoke are mutating your DNA, but where they mutate the DNA is just random. So if you're lucky and the mutations are not happening in key genes that control cell cycle regulation, then you may never develop cancer. But if you're unlucky, even with a relatively small dose of carcinogen, if you hit the quote unquote wrong genes, then you will likely get cancer. So typically at low exposures, the chance that one of those genes that's involved with cell division, cell cycle regulation, some of those key genes that we think of as potentially cancer causing, the chances that those genes are going to be affected is quite small. At higher doses, chance is much greater. So really it's all about minimizing your exposure as much as possible. And so in terms of the history, how did we first realize that chemicals could cause cancer? Unfortunately, in most cases, we don't realize that chemicals are carcinogens until after they've already caused many cases of cancer. And historically, really, we first started to identify these by seeing groups of individuals that had sort of rare, unusual cancers that also correlated with kind of unusual exposures. So we could take this all the way back to the 1700s, the mid-1700s. This London physician, John Hill, noticed a group of patients that all were developing nasal cancer, which is kind of unusual. And it turned out these people were also routinely using snuff, which, if you're not familiar with that, is basically an inhaled form of tobacco, basically like powdered tobacco that you would inhale. So they were engaged in this somewhat unusual activity of inhaling this substance, and they were developing a cancer in that same tissue exposed to that substance. Around that same time, it was noticed that there was quite high incidence of scrotal cancer, again a pretty rare cancer, in chimney sweeps, and chimney sweeps were usually boys. They had to be small so they could fit down chimneys and literally, sweep them out with brushes. But they had this unusual exposure, and that they were, being basically completely covered head to toe with soot and So they were able to make this correlation. This exposure to soot on the skin is causing this rare incidence of cancer. And something more recent, in the 1900s, we saw high rates of skin cancer in the coal tar industry, etc. So it was those kinds of correlations that kind of brought about this realization that yes, exposure to certain chemicals can greatly increase your risk of cancer. Another example I'll come back to in just a little bit is bladder cancer in the aniline dye industry and in the 1900s. So there's a few trademarks of chemical carcinogens that I wanted to mention here, and I'll come back and talk about these in a bit more detail at the end of this section. But one is there's typically a long delay between initial exposure to the chemical and the development of that particular cancer. We already talked about the fact that you see that to be the case with exposure to the chemicals in cigarette smoke, and development of lung cancer. It may take 20, 30, 40 years for cancer to develop. You almost always see organ specificity, right? We could stick with the lung cancer example, in that you're inhaling cigarette smoke into your lungs, and of course those lung cells are the ones that are going to take the brunt of that and most likely become cancerous. Although that being said, smoking does increase your risk of many different types of cancer as well. And there's almost always a dose-dependent response in terms of the ability of chemicals to cause cancer. So higher dose, higher risk, more likelihood of developing the disease. I also wanted to talk about three main sources of exposure to chemical carcinogens, right? So where are we being exposed to these things? And one key finding, we've seen this throughout history, is occupational exposure, right? So if you work in an occupation where you are handling or exposed to somewhat unusual chemicals that maybe the general population might not be exposed to, or you're being exposed to these chemicals at higher doses because of your occupation, it may put you at higher risk. Of course, we discussed the fact in the previous chapter that our diet is basically a mixture of chemicals, right? So we're constantly being exposed to chemicals, both natural and man-made, in our diets. And then lastly, we kind of say more broadly environmental, outside of our diet or occupation, just going about living our lives in the world, we are exposed to chemicals in the air that we breathe, and other things that we come into contact with in our day-to-day lives. So I want to go through these three again. Let me go back here: occupational, diet, environmental. Just briefly expand on those three. So we'll start with occupational. And what you're seeing here is a list, pretty long list of chemical carcinogens where the human exposure is primarily occupational. And so, these tend to be more like industrial type chemicals, but there's a couple that I'll give you a little bit more information on here. Asbestos, right? So if you are a construction worker, you may be exposed to asbestos. And then 2-naphthalamine, which is used in the aniline dye industry. It's linked to bladder cancer, asbestos with lung cancer and mesothelioma. So because historically we were seeing these groups of workers, developing these somewhat rare cancers because of their occupational exposure, this definitely contributed to the creation of OSHA, the Occupational Safety and Health Administration, in the early 1900s, which helps, regulate and protect workers against these types of exposures. So just to zoom in on those two examples, aniline dye, I mentioned, this was a purple dye. It was really popular in the clothing industry in the late 1800s and into the early 1900s. You can see its chemical structure there. And this dye industry found that a closely related compound called 2-naphthalamine, you can see here, was a good starting material for synthesis of aniline purple, and a number of other dyes as well. But unfortunately, this 2-naphthalamine was found to cause bladder cancer in workers that were exposed to that particular chemical. And you can see here, it follows that kind of hallmark delay between exposure and development of cancer. So you can see percent of individuals with bladder cancer, and then years after initial exposure. So you've got the red line less than 2 years, blue line 3 to 4 years, green line more than 5 years after, right? So for sure, the more time that's passed between that initial exposure, the more likely you are to develop cancer. And I'll probably mention this again, but why bladder cancer? Well, in this particular case, the 2-naphthalene chemical is being metabolized in the liver if you get it into your body, and it's trying to excrete it through the urine after it's metabolized, so it concentrates in the urine, hence the bladder cancer. Asbestos, I don't know, you guys are young enough, you may or may not be familiar with asbestos. But it has been a major cause of cancer deaths, really around the world, but in the U.S. in particular. And it was used very extensively in construction materials all the way from the 1940s into the late 1970s. So I'm not going to get into this movie. It was basically a promotional movie from the 1950s about asbestos. But it's these very fine fibers that are, fireproof and just have this incredible insulating ability. So if you have an old home or building, if you go down in the basement, you may see some of the pipes or ductwork with this kind of insulation around it. That most certainly contains asbestos. It was used in, tiles for ceilings and floors. Primarily because of its toughness and its insulating abilities and fire retardant capabilities as well. But if you look at asbestos, if you were to break open some of that insulation, look at it on an electron microscope, you would see that it basically is this dust consisting of these tiny, tiny, tiny microscopic needle-like particles, right? So just coming across asbestos insulation is no problem. It's when it's being disturbed or torn out, really, that you would risk exposure. Maybe if you worked in a plant producing it, I suppose that could be an issue as well. But these particles are so small, they become airborne and you can inhale them. And then they get lodged in the lungs, and they can get very deep into the lungs, into the the little bronchioles and alveoli there, and you can see the asbestos fibers lodged down here and just kind of poke through, and some of them will actually work their way out into the chest cavity and embed in the mesothelial layer of cells that surrounds the lungs, hence this common occurrence of mesothelioma and or lung cancer associated with asbestos exposure. And basically what happens is those little needle-like particles just lodge there and stay there, and it just causes chronic, long-term irritation. And there's all kinds of examples. We'll get into this when we talk about in the chapter about infectious diseases and cancer. But anything that's causing long-term inflammation and irritation is typically triggering and inducing cell division type pathways and can help contribute to development of cancer in many cases. So luckily we figured out asbestos was a problem, and it started to be regulated in the late 1960s. So we don't use asbestos in any new materials, although there are still older buildings around that do still contain asbestos. So it's believed death rates due to asbestos peaked in 2010. So hopefully we'll continue to see that decline, but it's still big enough of a problem. You may have seen on TV, sometimes you'll see like the commercials that are promoting these kind of big class action lawsuits related to mesothelioma deaths. And I don't know if you ever watch any of the like home building renovation type shows, but occasionally on those you'll see them come across a building that has asbestos, and when they do the removal now, it's really quite a impressive scene where they have to basically line everything with plastic and pump air in, so there's like a positive pressure so these particles aren't escaping, and the individuals that are going in to actually do the physical removal are, essentially dressed in like hazmat suits with full respirators, and it becomes it becomes a huge ordeal. So you might get the idea, talking about exposure to chemicals and increases in cancer rates over time, that maybe more industry, right, as our country has industrialized, maybe that's contributing to this increase in cancer in the general population. But we really don't think that's the case, because although there has been this massive increase in industry over the past 150 years, really the only cancer that we've seen a very dramatic increase in is lung cancer, and of course that's due to cigarette smoking rather than industrial exposure. So yeah, there are these small pockets of people that work in industrial settings that may have been unfortunately exposed to some chemical carcinogen that do have a much higher cancer risk, but that's a small number of people. So the population as a whole, we're seeing cancer rates for the most part remaining pretty flat, or in some cases declining due to better treatments or detection methods. All right, so let me come back into these three main sources of exposure to carcinogens. We're just talking occupational. The second one is diet. We go into that. Then we had environmental. So just to talk about diet, I mentioned in the previous chapter that it's because our diets are so complex and there's so many chemicals that we're ingesting, every day, it becomes pretty hard to tease apart those that may be posing the greatest risk. And I had mentioned saccharin on an earlier slide, so this is a good example, right? So this is a sugar substitute, right? You see it, still see it in Sweet and Low in the pink packets on basically every restaurant table in America. I can't remember, this was in the 80s, I want to say. Animal testing showed that saccharin was carcinogenic. It was causing bladder cancer in rats. So, of course, based on that data, the U.S. government tried to ban it, and I think it was actually banned, or at least deep into the process of being banned for a period of time. But as they dug more into the testing, they found that this was a case where the physiology was a little different, and for whatever reason, rats seemed to be more sensitive to saccharin than other lab animals, and certainly than humans were. And coupled with the fact that it was taking a very high dose, even in rats, to cause cancer, the data just suggested that, okay, this isn't really a big risk for humans at all. So, of course, as I said, now it's it's back on the tables and in people's coffee and iced tea. I know prescription drugs aren't really truly part of your diet, but they are something that we ingest intentionally. So I wanted to mention these in this same section as well. And unfortunately, there are examples of prescription drugs that have been linked to cancer. One famous one was DES, which was a synthetic estrogen that used to get pretty heavily prescribed to prevent miscarriage. And this was a really interesting example because it turned out, well, the women that were taking DAS actually didn't seem to have an increased cancer rate. But if they had daughters, those daughters whose mothers had taken the drug had much increased rates of vaginal and breast cancer. So, of course, once that was realized, then that drug was pulled off the market. Another common example of drugs that are still on the market and that are known to cause cancer or increase cancer risk pretty dramatically are some of the immunosuppressive drugs. But of course, those are typically being taken in situations where you're having an organ transplant, right? And you want to try to prevent rejection of that organ. And the idea is, well, okay, the increased risk of cancer is probably worth it, because I'm going to not reject this organ and not die from, liver or kidney failure, or whatever the case might be. So then that leaves us with environmental exposure. And as I said, that's really everything else that's not occupational or that we're not intentionally ingesting. And of course, that again is very varied, right? We're exposed to all kinds of things in our normal environment. So it becomes difficult to tease apart, as with the case of diet. But one thing that's clear for sure is that our main exposure to pollutants and carcinogens, sometimes those are one and the same, is undoubtedly indoors, right? Inside buildings. Um, so this is data from people that are equipped with little portable air quality monitoring devices. And you can see the little graph up here is looking at toxic, volatile, organic chemicals. The bottom graph looking at a variety of pesticides. I think pretty much everything on this list is shown to be carcinogenic, at least at some level. And you can see the orange bars here are inside and the blue bars outside. So, pretty much across the board, you're having much higher exposure inside. So what does that mean? Does that mean we should all just live outside in tents so we're avoiding this exposure? Well, obviously not, right? That's not feasible, not practical. And, even though your exposure is increased inside to these specific chemicals, really the heart of the matter is, okay, well, what does that translate to into real health risk, real increased cancer risk due to that exposure? And I think, we may not be able to answer that at this point. It may be unclear. But anyway, spending some time outside is probably going to be good for you for sure. Okay, so coming back to these trademarks of chemical carcinogens, we had this long delay between initial exposure and development of cancer. We had organ specificity, and we had dose dependence. And so I just wanted to go through these briefly, just give you a little bit of more information on all three. And so, one question you can ask related to this kind of ties in with the idea of this gap in time between exposure and development of cancer is, well, in most cases, most people are being exposed to low doses of these chemical carcinogens. So what are the risks of those low dose exposures? We know high doses clearly a problem. They're usually pretty easy to identify because you get high rates of cancer in the small group of people that receive those high doses. But those low doses are going to be a lot more difficult to identify, and in some cases they may still be potentially very dangerous, right? Especially if you're thinking of it in terms of the long haul over a long period of time. And this kind of begs the question, well, what is an acceptable dose or an acceptable low dose? And you might be tempted to say, well, 0, right? I don't want any exposure to carcinogens, but that's just not really practical. So you're going to be exposed to carcinogens no matter how you choose to live your life and what choices you make. So we really want to be able to say, well, what is an acceptable dose? And there's some weird examples out there, like this chemical dioxin. I'll come back to it in a minute, but at high concentrations, dioxin very clearly is linked to cancer, seems to be a potent carcinogen. But at lower doses, all the data shows it actually decreases your cancer risk, or at least in all the animal studies that have been done. And I'll come back and talk about how that could be the case. But there are some sort of unusual examples like that. So this ties in with something we talked about in an earlier chapter. We were discussing animal studies. It can, in some cases, be pretty difficult to estimate what the risk of low-dose exposures. I talked about the fact that when you do animal studies, you will typically do a few higher doses, right? So you may see this pretty linear relationship up here in your animal studies, and let's say this is the typical quote-unquote low dose that humans might be exposed to. It's tempting to be able to draw that line straight back since everything was linear up here. But there are many instances of these kind of threshold dosages where if you're below that particular threshold, your cancer risk is very low, but as soon as you cross that particular dosage, that particular threshold, then your cancer risk jumps way up. Right? So that complicates things and makes it more difficult to estimate. So while we're on this topic, I just wanted to bring up these three different models talking about this dose-dependent relationship. And of course, there's the linear model, where as your dosage increases, so does your cancer risk. There's the threshold model that I just talked about, where cancer risk is very low until you cross the threshold, and then it increases, at the very least linearly, but maybe in some cases it may jump up exponentially. And then we have the hormetic model, and this is where I'll tie this back in with dioxin. So in this is related to the threshold model, but in this instance, in the hormetic model, if you're below the threshold, your cancer rates or risk is actually reduced, but then as soon as you cross the threshold, again, it jumps up. So, what's going on in these instances? Why is it, what's so magical about that threshold, I guess would be the best way to say it. Why do cancer risks increase beyond that particular point? And there's really two main ways of thinking about this. So one is that at higher doses, carcinogens will actually start to kill cells, cause tissue death. So it could be, once you've crossed that threshold and gotten to a dosage where you're killing cells, then that's stimulating cell division, right, to try to replace those missing cells. And anytime you're activating those cell proliferation pathways, that you're already one step kind of down that pathway towards cancer. And related, but sort of tied in with this whole idea, is a lot of these chemical carcinogens will work by damaging DNA in some way or mutating DNA. And it may be that at lower levels, your cells have repair machinery that's able to handle that low level of damage. But as soon as you cross some threshold point, then the repair machinery is basically swamped and just can't keep up. And so then you start accumulating more and more damage and mutations. And so if you think of these two things possibly happening simultaneously, that really then becomes a recipe for development of cancer. All right, I will stop there and pick this up in part 2.